Hello, I'm Maxime Benalawa. I'm the Deputy Attaché for Science and Technology at the Consulate of France in San Francisco. I'd like to welcome you and thank you for joining in for the final panel of our week-long event on AI, Big Data, and Health. This panel is going to be about personalized medicine, and it's going to be moderated by Ashworth, whom I'll let start the conversation in a second. But before that, I'd like to thank again, because this is the last panel and my final opportunity to do so, I'd like to thank again the wonderful teams at QBI for organizing this event with us. It has been great work and a wonderful experience to work with you. I'd like particularly to thank Gina, Jacqueline, and Alexa for the wonderful efforts they've put in. And without further ado, I'll let Alan start the conversation.
Thanks very much, Maxime. It's delightful to be here. Bonjour or bonsoir, wherever you are in the world. I'm Alan Ashworth. I'm president of the UCSF Cancer Center, the Helen Diller Family Comprehensive Cancer Center here in San Francisco. And we've got an excellent panel of experts in precision cancer medicine. We have Dr. Sophie Postel Vinay from the Institut Gustave Roussy in the south of Paris. We have Dr. Larry Fong from UCSF and Dr. Alejandro Sweet-Cordero from UCSF. And I'll give them an opportunity to talk about their roles in precision medicine and challenges and opportunities. And that's what we're going to focus on. Just got a message to say that Maxime forgot to say that the Consul of France will give a quick conclusion speech at the end, so please stay on for that. What we're going to do first is I'm going to set the scene with a couple of slides on defining precision medicine. And this is a subject where you ask 5 people what it means and you'll get 5 different answers. And that's one of the things we want to try and clarify if we can today. Next slide, please. So this is an interesting perspective on precision medicine. So Hippocrates, 2,500 years ago, said it's far more important to know what sort of person the disease has than what disease the person has, which is not so far away from our definition of personalized or precision medicine, that the person, the nature of the disease that the person has are interrelated. And of course, our modern definition of precision medicine was articulated by then President Barack Obama at a launch of the Precision Medicine Initiative in the US in 2015, where he said the promise of precision medicine, delivering the right treatments at the right time, every time, to the right person. And I think that's a fair definition of where we are with precision medicine, although it's been extended now to include immune-directed therapies. They use the host system of the person to attack the cancer. So again, they fall in the category of precision medicine. Next slide, please. So if we define what traditional cancer medicine has been and continues to be for many people, that is the primary modality of treatment. The initial modality is often surgery. So one takes out the tumor physically. And then there's treatments that follow that: radiation treatment, burn out the residual cancer, or chemotherapy, traditional chemotherapy, a poison to kill fast-growing cells. And to be fair, most people still receive most of those modalities. But what we're moving to, and I would say we're in a transition period, is to understand the genetics of the person and the tumor. So what does the genome look like? What genes are altered in the tumor? And we know cancer is a disease of genes largely with genetic alterations. And then exploit that for targeted therapies. And I also talked earlier about immunotherapy, identify ways in which we can turn the host immune system on the cancer to try and use the existing body's mechanisms for rejecting the cancer. Next slide, please. So as I've said, two major themes: biomarker-driven targeted therapy. So one has a molecular target within the tumor, and we have a biomarker, some way of ascertaining which patients are going to benefit from the targeted therapy. And those two things go hand in hand. And then the second, harnessing the immune system to attack cancer. Next slide, please. So that's really all I wanted to say by way of introduction. I show you here what is the embodiment of our efforts at UCSF in precision cancer medicine, our lovely new building, which we opened last year. So I think you can turn off the slides now. Thanks, Alexa. So I'm going to ask each of the panelists to talk for just a couple of minutes, up to 5 minutes, on what their role in precision cancer medicine is and what their perceptions of challenges and opportunities might be. And then we'll get into questions in more depth. So I'm just going to kick it off by saying I'm the president of the UCSF Cancer Center, as I mentioned, and my role is to translate discoveries, help translate discoveries by our investigators into the clinical arena, and I'm also responsible for cancer clinical services. So I see both ends of that continuum. I'm also a researcher. So in the 1990s, I was involved in the discovery of the BRCA2 cancer susceptibility gene, and then went on to identify therapies, specific therapies to treat individuals with mutations, a treatment called PARP inhibitors that are now standard of care for BRCA mutation carriers. So through my work and involvement in this area, I've got a lot of insight into the challenges and travails of developing new agents. The whole process of the discovery of the gene to the approval of the PARP inhibitors took 20 years and probably should have been 5 or 6 years less because this is a tortuous process of drug development. Yet we now have a treatment that is quite effective for ovarian cancer and somewhat effective for other cancers with BRCA mutations. Really, the principle being that the mutation decides who gets the therapy, not the site of the cancer. So now I'm going to ask Dr. Postel Vinay from IGR to introduce herself and say a few words.
So hello, everybody. First, I'd like to thank the Alan Ashworth and the organizing committee for offering me the opportunity to participate to this panel. I think it's going to be a very interesting and challenging topic. So I'm working indeed at Institut Gustave Roussy in France. I'm currently the head of the phase 1 committee, which means that I work in a department which provides new drugs to patients. These are drugs which are administered to the patients for the first time or for one of the first time. And we aim, of course, at finding in which patient population this drug can be effective. So in such program, it's very important to have the characteristics of the tumor of the patient, which is exactly the aim of precision medicine, which is administering the right drug to the right patient with the right tumor and as well at the right time. So to me, this last thing is one of the biggest challenges of precision medicine. I've been involved in several precision medicine programs at the national and at the local levels. And one of the biggest challenges, to me, are having the right drug at the right time. Because drug accessibility is probably the most limiting factor at the moment, but this is something we can discuss later. Other big challenges are to address the resistance. It means the tumor can adapt to the drug and the drug does not work anymore. This is a big problem. Another problem is the training of the physicians, a challenge at least, so that they can understand exactly what is the profile of the tumor and how to choose the best drug. Another challenge, as Alan said, is to integrate everything together. Like what he called precision medicine is not exactly the same as immunotherapy, and how can we integrate both things together to give the best drug to the patients. So I think this is just setting the frame, and we can go into more detail later. And so I'm going to give the talk to anyone else who's following me. Thank you.
Thank you very much, Sophie. That's a great introduction, a great way to start. Perhaps I move on to Dr. Sweet-Cordero, who's a UCSF colleague who treats pediatric cancers, children's cancers.
Yes, so my name is Alejandro Sweet-Cordero. I'm professor of pediatrics. I've been a physician scientist running a research lab and taking care of patients for about 15 years now. And I was recruited here to UCSF about 4 years ago by Dr. Ashworth and by the group in Pediatric Oncology, really to develop our efforts in genomic analysis of pediatric cancers. And then more recently, the request of Dr. Ashworth to lead a program that he developed called the Molecular Oncology Initiative, which is really tasked with the, as Dr. Vinay was saying, the real-life implementation of precision medicine to help our cancer patients here at UCSF, and also referrals that we receive from around the country. And so I agree with what Dr. Gustave was saying in terms of some of the challenges that we face in our day-to-day practice in terms of applying precision medicine. Certainly, we've had very significant advances, for example, with sequencing. 20 years ago, when I was starting my research career, I remember being in the audience at MIT when Eric Lander announced the sequencing of the human genome, which was an effort that involved many hundreds of investigators around the world and cost over $1 billion. And now here at UCSF, we can sequence the cancer-relevant genes for a tumor for just a few $100 with the tests that we developed here. And that's a huge advance, no doubt, and it has allowed us to really, as Dr. Ashworth was saying, really think about how can we personalize the treatment so that we're no longer treating patients based on what we see under the microscope, but we're actually treating them based on what we see in terms of the genetics of their tumor. But we still have a huge number of challenges, some of the ones that were mentioned by Dr. Vinay, for example. Many physicians are not really trained to think about their patients this way, so we need to do a lot of education. We have a tumor board that we run here where we try to do that education and help clinicians interpret the information both from the tumor, but also increasingly more important is also what we call the germline or the normal DNA from the patient, which also strongly impacts on the course of their disease and also the risk that they'll develop secondary cancers, as well as potentially some chemotherapies or other targeted therapies that may be more likely or less likely to work for individual patients. So despite all the advances we made in science, the implementation of precision medicine is something that is an ongoing challenge. I think I also wanted to highlight one of the other things that were mentioned earlier in terms of access to drugs. Many of the mutations that we see in tumors are still not what we call actionable. So we know that they're important. We know that they're probably contributing to the disease, but we don't have any drugs for them. So there's a huge need for greater development of drugs, particularly for rare cancers that don't get as much attention as some of the more common cancers. And then access to drugs, I think certainly in our country, we're all well known for having a very dysfunctional insurance system, and so that's our claim to fame. And so that makes it quite difficult. Sometimes we have patients who we know would respond to a therapy, and yet we have no access to the drug. And that's certainly a problem in the United States, but I would argue that's probably a problem in most of the world, since in very few countries are these drugs widely accessible. So I think those are some of our ongoing challenges.
Thanks very much, Alejandro. That's terrific. I'm now going to move on to Dr. Larry Fong, who's a world-leading expert in immunotherapy, developing new immunotherapies, and in fact was one of the first people to deliver an immunotherapy, ipilimumab, to a patient. Larry, you're on mute.
Great, thank you, Alan. I wanted to also thank the organizers for having me on this panel. As Alan mentioned, my interest is on cancer immunotherapy. I'm also a physician scientist, so I lead a research lab as well as a clinical program, and have been at UCSF for 18 years. over the last 5 years, Alan had tapped me to lead and build out a cancer immunotherapy program within the UCSF Cancer Center. And what this program is focused towards is really delivering new immunotherapies to our cancer patients. And a lot of these treatments actually have potential risks and side effects, and so we need to manage them. And so we've built out a program that allows us to do this and really try to deliver treatments to patients that we think might respond to these immunotherapies. I think, you know, one of the big excitements around cancer immunotherapy is that when it works in a patient, it actually can work incredibly well. In that we have patients who have cancer that has metastasized or spread throughout the body. And usually with our conventional cancer treatments, when we get to that point, our treatments are really to try to shrink tumors and try to make the patient as comfortable as possible for as long as possible. But we don't really have treatments that can take away a lot, if not all, of the cancer. And with some specific immunotherapies, we now have patients where we've treated patients with immunotherapies over sometimes a very short period of time, like 3 or 6 months, and their cancer has gone away and has remained in remission now for 5, 10 years in some of these patients. And this type of response that's durable even while you're not receiving treatment is something unique to immunotherapy. And I think many of us in the field believe that some of these patients are indeed cured of their cancer. That's obviously a very loaded word in the field of oncology, but many of our patients who have these really dramatic responses go on to live their normal lives, and it's really gratifying to see that. And that's something that we would not have seen with our usual cancer therapies. The challenge with immunotherapy is that we've got these great responses, but it only happens in a minority of patients. And this is really, the need with regards to precision therapy is try to understand which patients actually can have these deep responses and which patients we actually may need to tailor other types of newer immunotherapies to try to get those patients to also have these great responses. And the challenge in our field is that we think about the genetic alterations that might be present within a patient or within their actual tumor as we've touched upon, but then you have to superimpose on top of that a person's immune system and how their immune system actually interacts with the cancer. And that just adds another layer or multiple layers of complexity. And so at UCSF, our program is really focused not only on treating our patients, but learning as much as we can from the patients we treat by doing deep immunologic profiling of the patient. That means understanding their immune system, not just in the blood, but in the tumor and in other areas of the body. And this is something, our hope is that as we really understand all these different elements and how they interact, we really can translate these clinical responses we see with immunotherapy to more and more patients. And so maybe I'll stop there.
I think you make a very important point there, Larry, about the linkage between immunotherapy and what we've traditionally seen as precision cancer medicine. That immunotherapy is dependent on the host genetics, which determines the host immune system in part. It's also the exposures that the body gets, as well as the genetics of the tumor, the presentation of neoantigens. We know that is incredibly important in response to checkpoint, immune checkpoint inhibition, for example. So more mutated tumors tend to respond better to those types of therapies. So I think we're seeing the fusion of those two areas. I want to start off by, taking a kind of naysayer's view. You're all proponents of this field and, invested in it. There are those that say, well, you're spending an enormous amount of effort on a very small number of patients. Actually, we're not shifting overall survival in most cancers. Maybe some we are, maybe lung cancer's as the first indications that there might be a change. But that it is marginal. We sequence all these patients, only a few respond. Even in immunotherapy, most patients with any given cancer won't respond. So let's just go around and let's have a balanced discussion for a little while on positives and negatives. Sophie, do you want to kick off?
Yeah, I think regarding precision medicine, it's true that there are a minority of patients who will benefit from targeting the alteration that we find at the moment. In our series, it's close to 20%, so it's still something. I think it's still important, because when you're in the 20%, you get the benefit. I think there are several things which contribute to not being perfect. First, as I said, it's drug availability. Sometimes we have the drug, we have a good drug, but it's just not available at the right time. So the patient won't benefit just because it's in a clinical trial and you need to have a slot at the right time when you find the mutation. So this does not occur. Or because, for example, in France, sometimes we get the approval later than in the US by the FDA. So EMA is always a bit, for some drugs, not all drugs, and I think olaparib is a counterexample, the one you developed, but there are very few counterexamples. So this is just something as well, which means that the timing is important. Sometimes as well, we need better drugs. We have drugs that we have what we call first, second, third generations of some drugs. And it's only the third generation which will bring the real benefit to patients. So this is also something we need to improve, which is having better drugs. And another thing is that we always talk about one mutation, but when you sequence a tumor, you will find several of mutations. So, and as you said, you also have the host immune system and everything which talks to the tumor. So we cannot completely dissociate both, even for targeted therapies. So I think knowing which one needs to be targeted and at which time, and whether you don't have another mutation which will have a negative effect on what we would call the driver mutation, that's something on which we need to really improve our algorithms. And that's the role of the molecular tumor boards. And maybe as well, artificial intelligence could have a role there in terms of like improving the algorithms of having always a better decision based on the profile of a patient.
Thanks, Sophie.
So
that's terrific. Yeah. Thanks. And yeah, I think your point about the using algorithms to triage molecular changes in tumors into actionable and non-actionable events is something that we're all very engaged in. I always joke that a molecular tumor board at the moment is 20 people in a room with their laptops open searching the internet, which more or less is the case. I mean, they're more or less experts, but then we see mutations in genes that we've never heard of, or we see mutations in genes that we have heard of, but we don't know what they mean. And I think it's the level of our knowledge base that is very limiting. Alejandro, you run the molecular tumor board. How do you see this area evolving?
Yeah, I think that some of the challenges that were brought up, I think, are critical. I think one of the big challenges, I think, is even the term maybe precision medicine is our biggest liability in a way, or in the sense that if we think of every patient as being unique and having a unique combination of mutations, then it's going to be, if you take that to an extreme, it becomes very difficult to learn anything about any other patient because everybody is unique, right? So at the end of the day, we have to put patients into groups, or sometimes when we talk about clinical trials, we talk about buckets. In terms of, we have to think of them as, we can classify them, but at the end of the day, we still have to put them into groups and see if that group responds better to one drug versus another. And so how do we do that, in the real world? How do we design these clinical trials that are based on combinations of mutations? I think is a major challenge and probably requires a very significant amount of collaboration. So I think that this kind of forum is very important in sort of helping us build those connections so we can think of better collaborations. But I think the other issue is what was brought up earlier also about combination therapy. So we know And we all like to think of cancer as, something we need a silver bullet for, but with maybe a very few exceptions in terms of immunotherapy, perhaps, and some of the PARP inhibitors. But for the most part, patients need more than one drug. And so how to combine those? When you start combining drugs, you also start combining and amplifying toxicities. So all of these drugs, even though we like to think of them as being very directed, pretty much every single one of them, including all the immunotherapies, has quite significant toxicities that that also become a challenge in clinical practice. So I think some of those are some of the things that we struggle with when we're trying to make recommendations for our patients at our molecular tumor board.
Thanks, Alejandro. Larry, in the field of immunotherapy, one of the issues that comes up in terms of benefit is also the cost. And that's the cost of long-term side effects that can be extremely debilitating and life-threatening. Colitis, blindness, diabetes, all sorts of things that that you may argue that it's better to have those and be alive, but it is a very serious set of potential side effects.
Yeah, I mean, that's something that, we're continuing to learn about. A lot of these immunotherapies, especially the immune checkpoint inhibitors, the way they work is by removing the brakes on the immune system so that immune cells that a cancer patient might possess can go on and attack the cancer. And that's how those treatments work. But the problem is, if you're removing the brakes from the immune system, certain patients may actually have immune cells that, rather than targeting the cancer, might target these other types of tissues where we see these side effects. And so as a result, the immune system attacks those tissues, as you mentioned. And there's a lot we need to learn in terms of understanding how this actually happens, and whether or not we can actually identify which patients are at higher risk for these immunotherapies. And we could think about, in that context, managing those patients in a different way, either by modifying the immunotherapy that they receive, or perhaps starting to use treatments that might dampen an immune response to prevent those side effects from coming on. This is an area where there's a lot of research going on now to really try to figure out the best ways to manage these patients. And it's one where, as you mentioned, now that we're having these patients who are long-term survivors, but now are impacted by these side effects, that we're really trying to refine our approaches in immunotherapy. But it's an area that we really need to make many more advances in our understanding to really, effectively avoid some of those side effects.
One of the other criticisms of high-tech approaches, precision medicine, whatever you want to call it, is that they amplify inequities rather than reduce inequities. So effectively, the rich get richer, that people can afford the treatments, get the treatments, and the disparities are amplified. And this is particularly in the US. It may apply in Europe as well, with whether you're treated at an academic center or you're treated at a rural center. So perhaps Alejandro, you want to kick off with how we should go about reducing the disparities in outcomes via precision medicine.
Yeah, no, I think that that's a huge challenge. I mean, certainly, in systems where not everybody has access to the same level of care, even here at UCSF, we struggle with that. For example, sometimes clinicians will order only the tumor, but not the germline DNA, in order to try to save patients money, which sometimes can be a reasonable thing to do, but also sometimes doesn't allow us to see the full picture. And so I think providing broader access and really being advocates for our patients, I think we all do it on a one-to-one level in terms of fighting for patients to get access to particular drugs when we see that they, for example, have a mutation that would be easily treatable with a mutation. We spend a lot of time calling insurance companies, arguing for access to drugs. But I think perhaps we should think about ways to do this more as a community, as advocates for patients in a more kind of systematic way. And I think that people do that, but I think there's probably more that we could do to speak for the need for greater access and more accessible care, regardless of financial situations.
I think part of the inequity comes from physician knowledge as well. That particularly out in the community, the knowledge of the value of genetic testing and advanced therapies isn't there, which means patients don't get offered such therapies. Sophie, in France, what is the situation with advanced treatments in rural parts of France, or those parts that don't have academic centers?
Yeah, so in France, there are several big academic centers across all territories, but it's true that the main disparity, I would say, is a geographical disparity rather than a financial one, because we have a completely different system. So in terms of genetic testing, there are national programs which are put in place. And for example, there is a very recent one going to 2025, where the aim is to sequence whole genome, whole exome, RNA-seq. That means a very comprehensive characterization of the tumor for more than 18,000 people a year. That's not only cancer, it also accounts for rare genetic diseases and cardiac diseases. But it means that that's a huge national effort. So in this context, it's true that the main question is how to bring this effort to patients who are far from big cancer centers. And for this, we have information platforms. We have, of course, networks between oncologists. But it's true that when we look at the recruitment of the patients, for example, in our phase 1 unit, you have 50% of the patients who will come from 100 kilometers around Gustave Roussy, and another 75% who will come from 300 kilometers around Gustave Roussy. The rest will come from the rest of France. So there are other centers than Gustave Roussy, of course, but it means that there is some geographical disparity, and it's very hard to bring that information to all the centers everywhere. And also because sometimes that's where we had an educational effort to make as well about the biology. Sometimes oncologists think that they have tested the patient because they have made a limited gene panels for the mutation they know. But they don't know, for example, and it's nobody's fault, it's just nobody can know everything, but they are not trained in knowing the differences in different types of mutations. And so they think they have done the right thing, they have tested the thing, but they have actually not. So there are lots of efforts to be done with regards to that.
Great. I just want to remind everybody that if you have questions for the panel, then please use the platform to ask them, and we'll try and address those questions. Larry, have you any thoughts on this area?
Yeah, well, immunotherapies are also just incredibly expensive. And, with some of our cell therapies, where, rather than giving something to remove the brakes from a patient, we might actually take cells out of a patient and re-engineer them so they actually can target the cancer cells. And these are now FDA approved in the United States for B-cell malignancies like leukemias and lymphomas. Just buying the cell product from the companies costs on the order of $400,000. And so this is just an enormous expense just for the cells. And then you actually have to treat the patient, bring them into the hospital, manage their side effects. And so you're talking about $1 million, when it's all said and done in terms of treating each one of these patients. And that puts a huge stress on the healthcare system, because as you might imagine, depending on how a person pays for their care, whether they're privately insured or depending on government insurance plans, what the health system gets reimbursed is actually very different. And that's something that, although, at UCSF, our mantra really is to care for all patients that come to our institution, you can imagine scenarios where applying that in reality may actually bankrupt the entire system to treat that small number of patients. And so this is something that we grapple with, as an institution, really, want, to do the right thing. And in some instances, that may mean that an institution takes on making those cells on their own so that we don't have to pay for, the $400,000 cell product. But I think, the other component is really thinking, about how we can better select, who really derives, benefit from these particular therapies. I think, reflecting on what, Sophie has relayed, in the United States, we absolutely have a geographic disparity. And, in our, immunotherapy program, we have patients coming from adjacent states and distant pockets within California where, the types of therapies are just unfortunately not available there. And we know we're only capturing a subset of the patients who probably would benefit from those therapies. And, it's just getting to the medical center, represents a challenge for a lot of these patients. And then, the final part about disparities that I just would want to, throw out there is, there are, patients who are uninsured, but there are actually a lot of patients who are underinsured. And that gap is actually a huge challenge. If you have no insurance, there are actually some safety nets in place. And for instance, sometimes companies will have patient assistant programs so that they can get you the drug. If you're underinsured, you kind of fall into this hole where you might not actually qualify for that assistance program, but yet your insurance does not adequately cover these very expensive treatments. And so that's another challenge that we face. And so it's on multiple dimensions that unfortunately can impact a patient's access to some of these different therapies.
Well, thanks for bringing up those points about reimbursement and insurance, Larry. As the person responsible at UCSF for the finances of the clinical cancer operation, it's something I worry a lot about. As Larry said, we don't send anybody away at UCSF, regardless of what their insurance is. But that creates, some significant issues. What do we say to the parents of a child who has a relapsed leukemia, where the cellular therapy has an 83% response rate and the responses are durable, yet the treatment will cost $1 million? Well, our answer is yes, we will treat you, but can we afford to treat everybody who comes if the entire state comes? And that's the issue we've got about how to equally distribute the costs around different organizations. I wanted to touch on the topic of children's cancers and the value of precision medicine there. What we've seen with children's cancers, and particularly leukemias over the years is great advances through combination treatment, through subsetting of tumors. And now we're seeing the advent of precision medicine there. And I know you've dealt with directly, Alejandro, cases where this has made a huge difference to the child.
Yeah, absolutely. We've had some amazing success stories here at UCSF, and of course many other institutions have as well. Some of those we've published on in papers where we describe what we call sort of N of 1 responses, when you have one patient who's had a dramatic response, and those are actually helpful to see because other patients might have a similar profile and might respond similarly. So we have an example that we just published a couple of weeks ago of a young lady who had a very dramatic response to immunotherapy and was essentially cured from a metastatic cancer, as Dr. Fong was describing earlier. So I think there is a huge potential in pediatric cancers, particularly because these are very rare cancers that are oftentimes not very good subjects for large clinical trials. And so, understanding the individual alterations in a patient sometimes is really the only option, because you're never going to have enough patients to be able to do a clinical trial for a very rare disease. Another place where we've seen precision medicine and particularly sequencing be extremely helpful is something that might be surprising to the audience, which is just the diagnosis. So we oftentimes think that diagnosis is something we're very good at, and we've been doing it for a long time. But a lot of these rare cancers can be quite difficult to diagnose based on purely histological or microscopy-based approaches. So we, for example, have an effort here funded by the Cancer Center with support of Dr. Ashworth to try to expand our genome sequencing to Mexico and help the Mexican government develop a genome sequencing effort for children. And one of the main focuses there is not really therapeutics, because we know that access to the drugs is going to be quite difficult in a setting with limited resources, but helping in many countries, helping develop a system to be able to better diagnose patients in situations where, for example, many small hospitals may not have access to very sophisticated pathology techniques that would allow the diagnosis, or even having pathologists with expertise in pediatric cancers having access to a sequencing platform such as the one we've developed here, but accessible in many other places, really can help diagnose these cases, and that can have profound implications not only for the proper treatment, the proper surgery, but also for prognosis and for many other aspects of clinical care. So yeah, so we would definitely feel like precision medicine is transforming how we think about pediatric cancers, and I think that's only going to continue in the years to come.
Yeah, and that, it's very important that, in the US and Western Europe, that we're having difficulties in rolling out precision medicine equally across our countries. It's very significant challenges. So that does raise questions about how we disseminate through the rest of the world as well, where there are huge limitations on resources. Parts of Africa, for a long time, it was difficult to get 5-fluorouracil, for example, as a standard drug, and then one despairs at the prospects of trying to put DNA sequencing in place. Yet the technologies are advancing all the time, and I think if we've got the will to do it, it can be implemented and can make a difference. Sophie, we've seen a lot of, I know you see a lot of patients with lung cancers, worked on lung cancer. We've seen survival rates increase quite a bit over the last couple of years, and really rather dramatically, actually. And some have questioned whether that's anything to do with precision medicine or some other effect. But it has coincided with the implementation of both precise targeted therapies to genetic subgroups and immunotherapy. So what are your thoughts on lung cancer?
So I think it's a very good example, because the answer is it's due to both, basically, and both don't work in the same subset of patients. Although, of course, we are trying to make immunotherapy work in the patients which currently benefit from precision medicine. But typically, some patients have very specific genetic alterations and will benefit from small molecules and will not benefit from immunotherapy, at least currently, from the current immunotherapies that are what we call immunotherapy in the classical way, checkpoint blockers. But others will benefit from immunotherapy, even more in combination with some conventional chemotherapy. So the benefit is even from both approaches, and they work in different populations. And I think now the biggest challenges will be how to address resistance to these treatments, because as Dr. Fong said, some patients will respond fully and beautifully to the treatment and will be cured. But also there is resistance to immunotherapy to, I think, a much more important extent than what was initially anticipated. And this is probably a major challenge at the moment. And then the second challenge will be how can we combine, at best, drugs with immunotherapy so that we can be effective without being too toxic. Because, as you said, we have had huge improvement with immunotherapy, but we also had big disappointments because of huge toxicity of some combinations with no improvement in efficacy. And I think this is very important, as Dr. Fong said before. The management of toxicity is a major challenge. because the management of the toxicity clinically is very different from the management of the toxicity of other anti-cancer therapies. And you took the example of the diarrhea. This is something that the mechanism is different with immunotherapy than with conventional cytotoxic therapy. And you can make it worse if you don't treat it properly. So, for example, at Gustave Roussy, we have someone really good, which is Aurélien Marabel, who developed a national panel. And every 2 weeks on the Wednesday afternoon, we have discussions with specifically for immune-related toxicities. How can we manage them at best, and so on. And I think this is the kind of national effort which has to be done for precision medicine, has been done for toxicity in some specific settings, and probably should be even broadened because we all face the same challenges, I believe.
Thank you, Sophie. Larry, I wanted to ask you a question about cellular therapies, which, as we've alluded to, have been really quite successful in children's leukemia and somewhat in adult lymphoma. We've seen no cellular therapies that have shown much efficacy for solid tumors. What do you think of the challenges there, and how are we going to get these therapies working for solid tumors?
Yeah, I mean, that's really, a huge, challenge for the field. As you might imagine, if you've, engineered an immune cell and you give it back to a patient, if the cancer cells are circulating, like in a leukemia, for those immune cells to engage, it's a lot easier than asking that same cell now to actually go find a tumor, infiltrate the tumor, and then be able to kill it in that environment. And I think, one of the big challenges now is really understanding how we can get these cells to actually be able to do that, find the cancers and go into actual solid tumors. But the other challenge is when we think about these solid tumors, those are very inhospitable environments for immune cells. And, cancers have developed ways to avoid the immune system from attacking it. And many of these mechanisms are probably also operating on these engineered T cells. And so, over time, we've, made some progress in understanding what some of these limitations could be, but we really don't understand the full scope of that. And I think, that's going to be really, one of the big barriers, in terms of developing these therapies for solid tumors. That said, there has been some progress that's been made with certain tumors. Multiple myeloma, which is, a disease of, hematopoietic cells, but those cells reside in a bone marrow niche, which actually has some of these immunosuppressive factors. These therapies are now showing activity, but we're also seeing resistance. And we're really looking at, by these patients that we're treating, as in some ways, opportunities to provide insight into how we can armor these immune cells or engineer them in ways that resist these resistance mechanisms. And so this is something that, at UCSF and at many of the leading centers, around the world, we now look to treating our patients and seeing who responds and doesn't respond as actually the laboratory, as opposed to thinking about, what goes on in, each of our own respective laboratories. In many ways, the clinic has become the laboratory to address these questions.
Thank you, Larry. So we're coming up to the end of our discussion. It's gone very quickly. So I just we've got a couple of minutes before I ask the Consul of France to join us. Do we have any closing remarks? Maybe I'll just ask you to do a crystal ball. Where will we be in 10 years' time with precision medicine? Will we be still fighting the niche, or will we be mainstream? Sophie.
I think, well, I hope, but I think also that there will have been great progresses in 10 years' time. Probably we'll get much more data and much better understanding. as well as better drugs. I think now as well there are international efforts to share data, and this is really a major thing, because it's only, as you said, we will go further and further in the tumor biology. It means that every patient will become even more unique, and it's only by sharing data that you will be able to get to this kind of granularity, so that you can really give the right treatment to the right patient. And as well, I think there is a really nice synergy which is developing between immunologists and more traditional precision medicine oncologists. And to me, this will be the key in obtaining long-lasting responses in tumor in patients and fighting cancer. Yeah.
Thank you. Alejandro?
Yeah, no, I have no doubt that we're going to continue to make significant progress. I think if you look at where we were 5 years ago versus where we are today, it's actually quite amazing. And so I'm very optimistic that the precision medicine is here to stay and will continue to be a major factor in our ability to improve the lives of our patients and hopefully cure increasing numbers of them and certainly prolonging life. So I think that's going to happen. I think some of the issues that we talked about today are critical in terms of access, in terms of equity, in terms of financial access, all of those things. But at the end of the day, I think we are making progress, and linking genetic mutations to drugs is going to be more and more something that we're getting better at doing.
Larry?
Yeah, I would absolutely agree with my fellow panelists. I mean, reflecting on the last 5 years and where we are today, it's really amazing to see how treating cancer patients has changed significantly. And I never thought even though I've been focused on cancer immunotherapy, I never thought I'd see the day where patients with metastatic lung cancer, as the lead-off treatment for most patients is an immunotherapy. And I think harkening back to what Alejandro highlighted in terms of the sequencing of the human genome, that was a huge advance in terms of giving us a roadmap in terms of understanding, just knowing what the landscape was. I think, we're seeing a similar advance with regards to cancer, where we've really are developing these roadmaps to understand what is wrong with cancer and what is wrong with the patient. And as we really extend our understanding so that we have a better view of that landscape, hopefully that will provide us insights into thinking about precision medicine on a much grander scale than the way we're practicing it today. And so, that's, I think, will be the excitement to see that unfold over the next few years.
Well, thank you, all the panelists, for their comments and for their optimistic approach to this subject. I share the optimism. I think we're all frustrated that things take longer than seems necessary. And one looks back 5 years, 10 years, 20 years ago, my God, we're in a different world. And hopefully in 10 years' time, the pace of accelerated pace will be even more. So I'd like to thank the audience, thank the panelists. Now we have time for some remarks from the French consul. Thank you.
Good morning to all on the US side. Bonsoir à tous pour ceux qui sont en France. I'm very honored to make these closing remarks at the end of these French-American Innovation Days. This seminar is the result, I think, of a great cooperation between QBI and the Science and Technology Services of the French Consulate in San Francisco. I have to admit, of course, I didn't follow all of your discussions, but I have the impression that in the past days went quite well, and that they were extremely useful to all. Along with the science and technology team of the consulate, Jean-Baptiste and Maxime, I want to thank the QBI team, especially Jacqueline, Gina, and Alexa, for their dedication and hard work in organizing this science fair. We are, of course, grateful, to all the panelists who kindly gave time to share their experience their expertise, with the public, and who made this event a success. I think participants got the chance to hear from entrepreneurs, from doctors, from researchers, from UCSF, of course, but also beyond, from many other public and private organizations in France and in the United States. I am not a scientist, but I am very aware of the importance of your individual and your collective work, especially as both of our countries are facing a terrible second wave of COVID. I would like to pay tribute to each of those at UCSF and elsewhere who are committed to improving the quality of care through innovation and through research. Data science, math, health, are fields in which we are on the eve of rapid and promising developments which have been outlined during this entire week. But these leaps forward will only be achievable through international cooperation, and I therefore hope that many new connections between participants will appear and develop and prove solid after this seminar. I heard Nevin say at the beginning of the week that he expects science to be on steroids for the coming months. I fully agree. After the November 3rd elections, we now have the opportunity and the political appetite also on both sides of the Atlantic Ocean to strengthen like never before the scientific ties between France and the United States. Let us seize this opportunity. Thank you so much for your attention.